I'm going to tell you results from part of my PhD research on a tracer experiment. So we're moving forward. We apply M15 and we trace over time. And this time we're focusing on mostly vascular plants, moss, and microbial communities. So we don't care too much about the trees. I mean, we do, but due to the size of the plot, we can't concentrate on the trees. And so just a brief introduction, boreal peatlands cover a relatively small amount of the land surface, about 4%, but these are actually significant carbon sinks in the global carbon cycle, accounting about 1/3 of the global soil carbon. And these are systems that are generally nitrogen limited. Broadly speaking, you can have two categories of peatlands, bogs and fens. And we're focusing on bogs in this talk because these are rain-fed primarily. They receive nutrients and water input only through precipitation. And that make it very ideal for our tracer experiment, applying nutrients. And bogs are relatively low in nutrients availability and very acidic and relatively low in species diversity compared to more groundwater-fed fens. In the boreal region, you have both natural and anthropogenic disturbances, including wildfire, permafrost degradation, and more recently human activities, including forestry, peat harvesting, agriculture, as well as oil and gas extraction. And all these activities could lead to increased air deposition, as shown by the previous presenter. So Alberta has about 16% of the land area covered by peatlands, and overlay on top of the peatland map, we have the oil sand map. And then as you can see, the oil sand region is mostly underlain by some sort of peatland. So up to 66% of the oil sands could be some sort of peatland. Oil sands can be peatland. Right now, it's about 100,000 hectares of the area has been approved for oil mining. 40% of that is dominated by some sort of peatland. So what are the environmental concerns related to the oil mining in the area? Well, obviously, mining will remove the surface vegetation, alter the hydrology and soil structure, and loss of net primary production, as well as the carbon sink function. More importantly, we're concerned about the air deposition from the emission of fossil fuel burning from the mining activities. And these numbers agree with the previous presenters. I'm not going to go over that. And overall, it has been shown in various ecosystems, increasing nitrogen deposition could lead to acidification of soil and the leaching of cations. As a result, you can also have increase in primary production as well as decomposition and overall decline in biodiversity. So more specifically, we wanted to look at the bogs, and we want to simulate the effect of increasing nitrogen deposition and want to use an M15 tracer to look at how the nitrogen is allocated among different components of the system. And we want to identify the key players in terms of nitrogen retention and cycling. And more importantly, we want to know how these boreal bogs will respond in the future scenario of nitrogen increase. To conduct the study, we stayed away from the nitrogen increased area, well, supposedly, but we found out later that one of the sites actually has elevated nitrogen deposition already. So, but overall, we stayed away from the oil sand region in the middle, close to Wabasca and Uticuma. We selected 4 mature bogs, and they have remained fire-free for at least 50 years. And then within each site, we selected 5 plots to apply nitrogen addition. So in year 2005, the first year of the field experiment, we added double-labeled M15 nitrogen ammonium nitrate to the plots in 4 dosages. Remember, the previous presenter mentioned 10 is considered a critical load in the system. So we chose a range of deposition concentration to see whether the critical load does exist in the system. And then in year 2006 and 7, we just applied a regular nitrate ammonium to continue the fertilization effect of nitrogen. And one of the plot received just DI water as a control. So each of the plot is set up as a 1.2 by 1.2 meter plot and subdivided into 4 quarters for sampling over time. And along the division line, we have crank wires inserted to measure sphagnum moss production growth. And then from one of the subplots every year in October at the end of the growing season, we collected all the above ground vascular plants and the litter. And then we take a soil core, 30 centimeter long soil core, section them into 3 centimeter sections, and use that to determine peat biomass, root biomass, microbial biomass, as well as tissue and tissue nitrogen and N15 concentration, and also calculate the overall recovery rates for N15. For the analysis of the data, we basically did mostly two-way ANOVA with time as a repeated factor. And then for different vegetation groups, they're analyzed individually because these are collected from the same plot, so they're not independent of each other. And then I'm going to talk about the recovery rates of N15 later on. So the first question we wanted to ask is how sphagnum respond, because these are the dominant peat-forming species in bogs. And logically, you would think when you apply the limiting nutrient, the growth of linear moss, in terms of linear growth, primary production, or tissue concentration should go up. And that's kind of our logical hypothesis. But that's not what we found. As you can see here, the y-axis is the linear growth in centimeters. X-axis are the nitrogen treatment groups. And then each bar is 3-year measurement. So what you have here is a significant two-way interaction. Basically, the response of linear growth to nitrogen is dependent on the time of your sampling. However, we do see a decreased linear growth in 2006 and relatively kind of recovered growth in 2007. So something is going on with time. And this pattern is consistent throughout most of the measurements. This is sphagnum net primary production and tissue concentration of sphagnum. What you have here, again, is two-way interaction. So the response is not just to nitrogen. It's also variable over time. And the only significant increase in terms of primary production is that in the first year, in a very high nitrogen load, and then that increase disappeared over time. Same thing here, except the significant increase in nutrient concentration is the last year of the experiment at a very high nitrogen treatments. So something is going on with the time. Keep that in mind. We did observe some response in terms of the stem weight and then stem count to nitrogen treatment. So these are significantly increased stem weight. And this is something we didn't expect. And it's kind of interesting that, keep in mind, linear growth didn't respond to nitrogen treatment. So these guys, instead of growing taller, linear growth, they grow heavier and denser to compensate for something that we don't know. But we do suspect, okay, let's revisit the hypothesis, sorry. Linear growth, no clear response. Primary production and tissue concentration did increase, but it's time dependent. So what's up with this interannual differences? And logically, we think about climate, something going on. And we know that drought can decrease primary production of sphagnum. And more recently, a meta-study conducted by this Limpens, they compared over 100 studies on sphagnum production across Europe, North America, and Asia. They concluded that overall high nitrogen loading negatively affect sphagnum production, and especially when you have also strong competition from vascular plants. And more importantly, they summarized that 1 degree increase in the air temperature is equivalent to 4 grams per hectare per year. Remind you, this is like 40 kilograms per hectare per year. It's like 4 times the critical load. So 1 degree increase will have very strong effect on production of sphagnum in response to nitrogen. So I think that's possible. So we went ahead and collected the data of the weather in the area. The green line is the regional average. And then the yellow and the red are the two study areas. The bottom one is the temperature. Up one is the precipitation. What you see here is a huge deficit of winter precipitation right before the growing season starts. In the meantime, you have much warmer winter temperature as well as very much warmer summer temperature during the growing season. So overall, we had significantly drier and warmer growing seasons during the study period, which complicated the response of moss to nitrogen alone. So what about the other groups of plants? I told you we're not looking at trees, but we did look at all these groups of plants. How important are they in terms of their retention of N15 that we applied? And does the pattern, do the patterns change over time? So that's the question we wanted to ask. So here we compared tissue and nitrogen concentration and tissue N15 concentration. So this is an indicator of how the uptake of nutrients. And we compared, this graph shows all plant groups, but the statistics are done individually on each plant group. So you have all the vegetation types, and then across 3 years, and then each bar is a treatment group. The first one is the control. And it's a messy graph, but I want you to focus on the two stars here. It's the small bog cranberry and the moss. And what you see here is in 2007, when we apply N15, you see a huge spike in their uptake of N15 right away. They're very speedy, very efficient. Same thing here, except it's much lower concentration. But over time, this spike disappeared. So it's telling us that these plants that grow on the surface has very extensive roots that permeate it throughout the pea surface, take up the nutrients right away, but they don't hold on to it as we apply more nitrogen in the following 2 years. In contrast, these two deep-rooted vascular plants, lingonberry and ledum, acquired their N15 much later over time, and they don't lose their N15 over time either. And then particularly in the stem, the woody tissue of ledum, you actually see over time increase. So this is telling us that these guys rely more, possibly rely more, these are just our hypothesis. We think that they rely more on the internal cycling of nutrients rather than direct access right away, right from the beginning. So this is something interesting that we kind of didn't think about when we were starting the experiment. So overall, how are these systems retain and recycle the nutrients? And to answer that question, we wanted to calculate the recovery rates of N15. It is basically you measure the N15 content in the treatment vegetation minus the control and divide it by the initially applied N15 to get a percentage of the recovery rate. So here you have different treatment groups, and then over a 3-year period, the kind of mossy brown is moss. The green is the root, and then above ground is lumped together into, because they account for a very small portion, into the green. And then the red, as you can see, are the ones that, the nitrogen amount that's not recovered in any of the studied pools anymore. What you notice right away is moss account for majority of the recovery as expected. These are the drivers in the system. They're very effective at taking nutrients. What's important to notice is that even within 3-year period, the amount of nitrogen, N15, that's no longer present in the studied column is significant. Over 80% by the year 2007 has gone somewhere from the system. So it seems like N15 disappeared pretty quickly, even in 3-year period. And this is particularly obvious in very high nitrogen load. The decline is pretty significant. So let's take a closer look at the above ground. We now divided them into different groups. I want you to focus on the red, the ledum, and lingonberry, the two deep-rooted species. And as you can see, when you have less nitrogen available, majority of them end up in ledum. But when you apply more nitrogen, most of them end up in lingonberry. So there's something competitive advantages of the two species. Once again, it's not something we expected, but it's really cool findings. So where did the missing N15 go, you ask? Well, I can't really answer that question because we didn't really look at all these pools, but we think the way that we apply the N15 is kind of a shock to the system. There could be a downward leaching right away because we couldn't just trickle nitrogen on the surface. We have to dump like a huge amount right away. Not all of them can be like absorbed right away. But there's also a possibility that dissolved organic nitrogen export somewhere, either downward or outward from the plot could happen, and also gaseous loss, and also transport through the trees. Remember, the trees have hyphae network, ectomycorrhizae hyphae, that could transport outside the plot that we collected from. And then also, clearly, we didn't cover all the vegetation species that we wanted to, in particular trees, and also the herbaceous plants that by the end of October, they're all dead and probably not there anymore, blown away. So in summary, the community response, N15 surprisingly moved through the system fairly quick, went somewhere that we don't know. Efficient species take up nutrients right away, but they also switch to more readily available sources. They don't rely on the internal cycling of N15 compared to the deep-rooted species. And we think the rooting depth and the growth form, and also their association with mycorrhizae, as well as their preferences for different form of nitrogen could play a factor in the differences. Overall conclusion, as expected, sphagnum moss is a key player in the system. And however, more interesting to us was that above ground plants differ in terms of their retention and recycling of nitrogen. And overall, the system is pretty leaky. When you apply a huge amount of nitrogen, a lot of them are actually not retained in the system. And we think climatic condition played a very complicated role in the response, and it's something that we need to consider in the future when we study nitrogen deposition effect. I would like to thank my committee members, other than my supervisors. These are Dr. Sarah Baer, Loretta Battaglia, and Michael Madigan at Southern Illinois University, Carbondale, with their help and support. Fellow graduate students, numerous field and lab assistants that helped me throughout the years, and then Sandy Vitt for support, and funding agency, SEMA, and plant biology at SAUC.